G'day, Dr. Carl, Shirtloads of Science, of course, University of Sydney again. And today we're talking with somebody who is working to save lives, improve the lives of those who suffer from genetic diseases. And you're thinking, well, they're pretty rare. Let me tell you the opposite. Genetic diseases impact more people than AIDS, which has killed 50 million people, and cancer combined. Cancer affects 120 million a year, kills 10 million each year. And it turns out that 10% of all adults and 30% of children in hospitals are there because of genetically related conditions. So please let me welcome to the microphone and to the camera Professor Leszek Lisowski. So that's a good Polish name, and I should be able to pronounce it better.
Thank you for having me.
So you're making the tools that can fix people who have genetic complaints. Now, I'll just do a little bit of a backup so people will see what you're doing. So the tools that you make are being used by people such as Ian Alexander?
Exactly.
So we had a podcast with him, and just to summarise it quickly, he was saying that with regard to a disease called spinal muscular atrophy, and forget the name, apparently, am I correct, the problem is that in their brain, the nerve cells are unable to make a certain protein. Is that correct?
Yes, there's a protein missing.
And so therefore, the brain can't tell the muscles to contract, and the kid dies often in the first year of life.
Yeah, so what happened in SMA, or spinal muscular atrophy, that because of that mutation, the cells, the neurons which connect the brain, we call them motor neurons because they connect the brain to the muscle, they start dying. And that ultimately, children lose control of all different muscles, including the muscles that are involved in breathing, and they die.
But in Australia, and I didn't know this, that in addition to the heel prick test, so every newborn baby, I used to do this when I was a doctor at the kids' hospital, you stab the baby in the heel with a little tiny needle.
Take a drop of blood.
Get a drop of blood, blotting paper, and you test for phenylketonuria. And if people are not familiar with that, look up the next tin of diet drink that you have, and it may well say, beware phenylketonurics, you can't drink this. And what happens, providing they know they've got this condition, they avoid certain, it's an amino acid, isn't it,
Yep.
they have to avoid. Providing they avoid it, their brain will be fine and they'll live a completely normal life, but they can't have this amino acid, so they can't have diet drinks, and apparently orange juice is another one
I think
as well.
so, yeah. I'm not 100% sure about oranges, but definitely the diet drinks is a big problem.
And I thought that was one thing that they did, and I thought that also besides analysing for that, we also analysed for thyroid disease, and blow me down, for spinal muscular atrophy. And then I was talking to my brother-in-law, and he told me that he, as a paediatric anaesthetist, would get something that you were and Ian Alexander actually made, and they would squirt this thing up the spinal cord of little babies. And so you need to be a skilled doctor. This is not a skill you get overnight, although he said it was. No, he just sort of said, it's not that hard. And then somehow, and use an adenovirus, and an adenotype virus. And am I correct in saying that adenoviruses are good at getting into some cells in the human body?
So the virus is called adeno-associated virus.
So there's a family of them, not just the adenovirus.
It's not adenovirus at all.
Oh, so it's not adenovirus.
It's a virus that was discovered as a contamination of an adenovirus. That's why it's called adeno-associated virus. It's called AAV.
And I'm very glad that you told me where I was wrong, because if you don't make a mistake, you don't make anything. But also it's good to admit your mistakes. Thank you for correcting me on my mistake.
Perfect.
So it's it's related.
Adeno-associated. So it's a virus associated with adenovirus.
And this virus apparently is good at getting into certain cells in the body, and you scoop out the insides. In fact, you're the one who does the scooping out.
I'm the scooper.
You're the scooper.
I'm the scooper.
You're the one who scoops it out, and then you say, okay, I have made you your tool, and then you pass it on to Professor Ian Alexander, and eventually it ends up at some little hospital in Newcastle. And so you squirt this virus, which is inside, instead of having its normal stuff, it's got is it right? It's got the bit of DNA to make this protein?
Exactly. So think of it as a virus is like a capsule of a pill.
Yep.
What's inside is a medicine.
Yeah.
And this is just the shell, the outside. Yeah. So AAV by itself, it's a human virus. Well, it can infect other animals as well. But about 70% of humans have been infected by it. Nobody has ever been sick. So it's not pathogenic.
Really? So it's just a really mild virus.
It's not pathogenic. We don't know of any disease it causes. So, but nonetheless, we take the virus, we remove pretty much almost everything from inside.
With a miniature ice cream scooper.
Exactly.
We'll
Put it on the side for later.
Exactly.
Okay. And then we put the therapeutic DNA, which will be, let's say in SMA, will be the gene itself.
So SMA, spinal muscular atrophy, and how many rungs on the ladder of DNA would it be? Like 10 or 100 or 1,000 or 100,000 or a million?
So the AAV, because it's a shell, has a volume. So it's limited by how much you can put inside. So you can put about 4.4 kb, so 4.4 thousand nucleotides in a sequence.
So the DNA ladder of life is like a ladder with rungs.
Yep.
And we humans have got 3 billion rungs, but you can put inside this AAV, this adeno-associated virus, about 4,500
So
rungs.
it's actually very small amounts.
So how does squirting it into the spine, into the cerebrospinal fluid, how does it magically travel all the way up, and then how does it know which brain cell?
Well, so this is what the viruses do, yeah? So viruses have evolved to infect humans.
Yep.
Well, they evolved for many things, but they evolved humans, and when they go into human, they go to specific cells. So, for example, hepatitis C virus will go to your liver. HIV would go to your T cells. AAV would go to your liver as well. But there's types of AAV. So AAV is just a name, but there's many different types of AAVs. We call them serotypes.
I saw different numbers like 9 and 3 and 12.
So they're usually numbered, or there is a letter and a number.
Yeah.
So for spinal muscular atrophy, we use AAV9. AAV9 is actually very specific because for the drug that was developed for spinal muscular atrophy is called Zolgensma. Zolgensma.
And what is that? That's a drug?
That's the drug that cures spinal muscular atrophy. It's an AAV-based. It's the name of a drug.
Oh, so you call the whole package when you
when a doctor prescribes it, you prescribe Zolgensma. It's a brand name of the gene therapy that cures or treats.
And it comes in a little glass bottle or something, and you snap it open?
I think it comes in a little syringe and a vial, and you pull it in. And then Zolgensma is, my understanding right now, the only way it's given is through bloodstream. So it's given actually systemically.
Oh, because my brother-in-law was saying that he was...
Zolgensma has been tested, can be also given directly into brain or into spinal cord. But I thought that the approved drug, I think, goes systemically. So maybe your brother was involved in a clinical trial. I'm not an expert on that. But Zolgensma AAV9 is very unique because the human brain, which is where the cells would originate that we're trying to protect in SMA, human brain is obviously the most important. It's like, it's the hard drive of humans, like our CPU or everything in the same place. So it's protected. And we call it blood-brain barrier.
Exactly. The blood-brain barrier.
And the blood-brain barrier evolved to protect the brain from viruses, bacteria, large molecules. It filters, the nutrients can come in, but all the things that can be harmful, some toxins and other things, don't. So a lot of drugs don't penetrate into the brain.
So the blood-brain barrier wraps around the blood vessels, and it's kind of the feet of astrocytes. Look up astrocytes on Wikipedia, you'll have a lot of fun, and they're part of the glial support structure. Exactly. And by the way, just as an aside, people went looking at Einstein's brain and they found the same number of neurons as everybody else on a percentage basis, but more supporting cells. Go figure, sample size of 1.
Yep.
Okay, so you've got this blood-brain barrier which wraps around the blood vessels and stops viruses getting through. So how does your AAV...
Exactly, so it protects. So in this case, it becomes a problem because the barrier has evolved over millions of years to be very protective. But now the medicine is a virus.
Yes.
And the blood-brain barrier doesn't know it's a medicine.
The hollow coating is the delivery vehicle.
But it's part of the medication. So you cannot have one without the other. So for the drug that's inside, the DNA, to get to the neurons, has to pass through the blood-brain barrier.
Do you call it a drug or do you call it a genetic treatment? Or
no, it's called a drug or gene therapy. Or
it's a medicine.
It's a medicine, it's a drug. It's the same thing. I know drug has different meanings, but it's a drug in the medical sense. Yeah. So now when the Zolgensma AAV9 with the gene inside that's actually going to cure the child comes to the blood-brain barrier. The blood-brain barrier doesn't know it's a medicine. It's just going to stop it. Right. It's a filter.
Yep.
But AAV9 is the only one that we know of that actually has no problem passing through that membrane.
So it can go through the barrier.
Exactly. So if you give it systemically, it goes everywhere.
Systemically meaning into the veins.
Into the bloodstream. You get a bloodstream. It goes everywhere. But a lot of this crosses through the blood-brain barrier and can access the neurons.
Okay. Once it's crossed the blood-brain barrier, how does it know that it goes to not the visual cortex, not the hypothalamus, not the anterior cingulate?
Thank you for your question, because that brings me to the tool making.
Yeah, okay. So you are now an official tool maker.
That's how I declare myself. Ian Alexander will be the tool user. We have a joke about it, but I'm not going to make it on the video.
So you're the tool maker, so you'd have an icon. And these are tools that can save lives.
They are tools.
With regard to, and as it says here,
we call them actually devices. We call them medical devices.
10% of all adults and 30% of children in hospitals are because of genetically related problems. So you're making the tools that will help.
And what's important also, when I make a tool, let's say for spinal muscular atrophy, let's say, for a given disease, to deliver something to the brain, it's a tool. It's a platform technology. While Ian may use it for one disease, the same tool can be used for many other diseases as long as it's of the same organ. Yes. So I make something specific for the brain, it opens a lot of different diseases of the brain. If I make something for the eye, it can be used for many indications of the eye, and so on and so on.
But how does it know where to go in the
Exactly.
brain?
So now when you think about the pill, casing of a pill, like something that you swallow.
Yep.
Some pills dissolve in your stomach, some go to the intestines. It's the chemical composition of the casing that will determine where it breaks down. AAV is a little bit more complicated. So AAV is it's a ball, it's a shell. But it has a lot of little, it's a protein, so it has a lot of little extensions. And those extensions, we'll call them ligands.
Ligands.
And then cells have receptors. And they work like a key and a lock. They find very specific, it's extremely precise. So once it finds a correct receptor on the cell, it stays on and gets absorbed by the cell.
So the adenovirus you choose has got little bumpy things that say, I will be accepted by this sort of brain cell, but not that sort of brain cell.
For example.
And how do you find this out? Is it like experimentation?
It is a lot of experimentation in the lab. Sometimes we do it by design. So sometimes we learn enough about the cells that we say, okay, this cell has this lock on it, this receptor. Let's design something specific that's going to bind to it. That will be direct. That will be, by design. Sometimes I call it the spaghetti approach. You throw spaghetti on the wall and something would stick. Exactly. Thank you.
Okay.
But so if I say I want to transduce hepatocytes, the liver cells, as long as I have a hepatocytes on some kind of dish, I can take millions of viruses, and that's what we do. We make millions of variants, put it on the cells, which is the wall, and something will stick. So it's really the randomness of the process that then allows me to find the virus or the vector. We call them vectors when they're not viruses anymore. That binds to the hepatocytes. Later on, I would actually use it as a genetic tool, and I may study what receptor did I target. I don't need to know that. All I need to know is that I have a type of a AAV vector that is effective at transducing my cells, and that's enough to develop a drug with it. The mechanistic understanding is something that very often comes later, many years after.
Right. And just for the audience who perhaps might be very skilled in metallurgy, in biology, pharmacology, we have this concept of the receptor and the thing that goes into it, the agonist, or it's like a lock and a key. And if the key fits exactly in the lock,
very precise. It's like a key.
Things happen, and a cell might have 500 or something different types of receptors.
Many different ones. Yeah,
many different ones, and we just don't fully understand them. And apparently, one of the sodium pumps is associated with a family in Pakistan not feeling pain. Yeah. But we'll go back to that.
Yeah.
So, how many different diseases are you working with?
So currently in the lab we have about 12 in a pipeline, but actively pursuing about 6. The other ones we're trying to get more funding to develop treatments.
6 genetic diseases.
6 genetic
so
diseases.
were you involved with the SMA?
No, so SMA was done by a lab in US, by a company actually. Well, it started in academia, ended up being a small biotech company called Avexis, and then Avexis got acquired by Novartis. And
various things happen. So
exactly, but it's very similar technology.
You're very skillful at talking about the business side, so maybe we should ask how you got here. So I'm guessing with the name of Leszek Lisowski.
Yeah, you got better.
Yeah. That you grew up in Poland. Probably you had to learn Russian at school because it was still while it was occupied by Russia.
I made sure not to learn much. I was very resistant because
the more languages learned, the better. So,
well, I know, but I didn't think like that when I was a kid. It's because I was forced. Now I regret it, but because I was forced, I did everything not to learn it.
And so you were probably in a part of Poland that was not too far from the German border?
About 2 hours from the border, about 3.5 from Berlin. When I go home, I go to Berlin, actually. It's closest.
So how do you get to Australia? So what happened after high school?
Yeah, so actually after high school, I always want to be a doctor.
A medical doctor?
Yes.
So
I want to be a
pediatrician.
Are a medical
doctor?
No. So after, I know, I want to be, but I didn't.
So you became a PhD doctor.
Well, so what happened was I actually, after high school,
Yep.
I applied to college in US and I didn't get in. But I also applied to medical school in Poland, and it's very competitive, and I got into medical school. I went for 1 year to the medical school, and I was, it was my dream, so I was very happy. I was going to become a pediatrician. And after 1 year, I got a call from the school in US that rejected me the year earlier, that now they accepted me, and they gave me a full scholarship.
They gave you a full scholarship?
Full scholarship. So
does that cover?
It's like $50,000 worth per year, of scholarship.
So that covers the tuition and accommodation?
And food, everything was included. I didn't have to do anything.
All you had to do was study.
All I just had to do is show up and study.
This is one of my little prejudices. I think that study at university is a full-time job, and people should not have to work at jobs while they're doing this full-time job of being a student. Anyway, so you got accepted.
I sort of agree. But so then I had a choice: reject it, or what I chose to do was take a 1 year off. So I took a leave of absence, or we call it the dean's break. I took 1 year off, froze my medical school, went for 1 year to US. And I figured that I would learn English. It was a great opportunity, actually. After 1 year, I thanked them. I told them I decided to go back to medical school, and I wanted my scholarship to go to another student, and they were very supportive, and everything we said goodbye, and I went back to the school. So I went back to Poland, enrolled in a school. I was always enrolled, started my second year of school. And what I didn't realize is how much I'm going to be hated by the faculty. I know.
What?
That's when you realize that I live in a communistic country still. And I went to the evil America. And I
went to the forces of evil.
I started failing I was a little bit of a nerd, and I started failing classes. I started failing English class, for example, because my accent was American, not British. I mean, it was all coming out with excuses. But 6 months into my second year, Christmas break comes, I go home, my hands are shaking, I'm a mess, I'm stressed, because I became from a nerd. And after first year, I got a scholarship, like a scientific scholarship for having a very high average grades. I pretty much got
But now they were failing you.
And now I'm failing.
The same person working just as hard?
Well, I mean, medical school,
you have to work hard.
It gets, but I still was a nerd. Nothing has changed there. I just was failing, and I knew that I'm being targeted. And then a professor who was a young professor of psychology who came from outside, she actually took me aside and she told me that behind my back, the faculty is talking. And she said, you will never finish the school.
You will never finish.
So I knew I was being targeted.
So you could do all the study you like, but you wouldn't finish the last year.
I mean, I could probably finish, but and that's a discussion I had with my family. I could finish, but I'm probably going to be all gray and shaky hands. Like, what kind of doctor is... Like, I don't want to burn just because people have an agenda against me. But I was at home for Christmas. I was studying for an exam. That was right after. It was biochemistry. It's a tough exam, but I was doing my best, but nothing was going in my head. My head was, not in the right place. And I decided to try to go back to the US. So what I did is during the Christmas break, made a phone call. I was college. I made a phone call.
Plain old telephone system.
Oh, yeah, with a wire, and I had to and the numbers were rotating, one of those rotisserie things.
Telephone works.
Yeah, they do it, yeah. Reception is always good, on the landline. I called US, called the college. I told them that I apologize. I said, I made a mistake. Would you take me back? And they said, hold on, let us call you back tomorrow.
What?
They called me back the next day. I had a scholarship.
They called you back the next day instead of writing a letter there.
And it was during Christmas break. Yeah, so they gave me back my scholarship, housing, everything as how it was. On top of that, they allowed me to take placement exams because by now I have missed 6 months of school. So my friends from school, from my American class, are 6 months ahead of me. And some classes, you have to take one class to be able to take the next one. So I wouldn't be able to just easily jump in. They said if I come in and I take placement exams and prove to them that I know the 6 months, they allow me to catch up. So I will never have a 6 month break. I'll be back with my peers. And just the experience of how they treated a student, in contrast to what I was going on in my own country, because it wasn't that hard to convince me to move. It was really more about convincing my mom that she would not cry herself to death. Yeah,
that you had made the wrong decision.
That experience was pretty much when I told my parents. Both my parents said, that's it. You saw the outside. You don't belong in this country. You should go. So
what did you do at the college? Did you study for a bachelor's degree?
I studied for a bachelor in biology and pre-med.
Yep, and pre-med, and then?
I did still consider going to medical school in US, but in US it's very difficult to go to medical school and it's very expensive. And being foreigner, I couldn't take loans.
Yep.
So eventually I decided to go for PhD.
Yep.
It was a little bit of a mind shift because I was always aiming to be a clinician, and now I am a scientist. I never regretted that decision. I made it work. Obviously, I'm working in gene therapies. I get to help children in other way, maybe not directly.
Yeah,
exactly. So I found my way to get what I want to be, just a little bit different path.
So you went to Stanford and to the Salk Institute.
So I did my PhD. I did my PhD in New York at Cornell.
That's
and Sloan Kettering.
Yep.
And then I got
Sloan Kettering is good.
Yeah, it is pretty
very good.
I would
yes.
say, thank you. And then I got recruited by. Then I applied to different places for my postdoctoral training, and I went to Stanford. Yeah, I went to Stanford, and then I got recruited by the Salk Institute in La Jolla.
Is good.
It's really good. And then in the meantime, I met Ian Alexander when I was at Stanford still.
Oh, you physically met him in the USA?
I met him actually at the conference in Australia.
Oh,
so I won a fellowship when I was a postdoc, and the fellowship allowed me to have. I had my own travel budget, so I could go for a conference wherever I wanted. So when am I going to go? As far as I can and explore the country I've never seen. I came to Australia. And every 2 years, there's an Austro-Asian cell and gene therapy conference. Mm-hmm. So it's a local society. That year, it happened to be in Melbourne.
Yep.
I came to Melbourne. I met Ian. And then we met a few months after in US at the conference. And then after that, we will meet at different conferences, build a relation, publish a paper together, and recruited me to Sydney.
So now that you've got here, you mentioned that you're working on vectors against certain diseases. What diseases would they be?
Yeah, so since I moved to Sydney and I opened my lab at Children's Medical Research Institute, my lab is called Translational Vectorology Research Unit. So translational means we translate diseases, and vectorology, we make viruses. Yeah.
Translation. So you're translating
translation is taking
DNA.
something from discovery to clinical impact.
Oh,
so that's translation is the path towards clinical. Clinic. Exactly.
Yep.
Vectorology means we use vectors.
So you might say that a rat could be a vector for a disease, but in this case,
it's the opposite. So the vector in this the vector is a very broad. There's vectors in physics. Yeah, there's vectors is a generic word, but in gene therapy, it actually means the thing that carries. Carries your medical your medicine to the cells.
Rat might carry the plague bacteria.
Exactly. It's a vector of the disease to another host. Yeah. Right. So in a way, we're using viral vectors to bring the genetic cargo, which is the medicine, to the cells that need the medicine. I came across one disease I don't even I can pronounce it. CTNNB1. Yeah. Is that one that you're working on? Yes, that's actually the most advanced
one. At the bottom. Oh my God.
So what I specialize in, what my lab specializes in, is what we call rare diseases. Diseases are divided based on frequency. You have common diseases. Cancer is a common disease, although cancer is a group of diseases. Hemophilia would be, kind of a borderline. The fewer people per year are born with a given disease, the more rare it becomes. So you have a common, rare, ultra rare, hyper rare. As you can imagine, the bigger the disease, the bigger the market, the more attractive it is to commercial sector.
To the commercial sector.
Because that's what they want. And there's nothing wrong with that. They want to make money. That's their mandate. But that means that children affected by rare and ultra-rare diseases really don't get the attention from the commercial sector. And there's not much happening in that space, which means they fall through the fingers of the system, and then all of them would die. Or just don't have the life that they should. CTNNB1, those diseases are so rare sometimes. Some of the diseases we work with, there's 20 kids in the whole world.
20 in the whole world?
In the whole world. CTNNB1, we know of about 500. We know that the children are born at the frequency 1 in 50,000. So we know there's more children.
1 in 50,000?
So there's more than 500 globally. But they're not diagnosed, they're misdiagnosed, because CTNNB1 is not part of the newborn screen on that blood from the precursor.
And it seemed tragic where the child would progress past the milestones, and this would be heartbreaking for a parent, would then regress back.
Well, that's another type of disease. Those children in CTNNB1, it's a neurodevelopmental disease where children don't progress. They don't go back. They just don't progress fast enough. What is even more tragic, that as a parent, especially if it's not your first child, you know what to expect. And you very quickly notice something is not right. Some of those parents have to wait 7-8 years to get a diagnosis. Because the doctors are trying, sending tests. But by the time they figure out the correct test, or send it for genetic tests, but it depends where you're born, which country, which city. The medical system is quite different. Sometimes it takes many years for the parents. The child is obviously not progressing. He never went to school, never went to kindergarten, but they don't know what's happening.
Yes.
But then, and maybe Ian mentioned that, once finally they get diagnosis, they go to the doctor and the doctor will be like, okay, we got it. We know it's a CTNNB1 syndrome. We know which gene, we know exactly what's wrong. What's going to be the next question that the parents are going to ask?
Can you fix it?
What do we do next? Yeah, and in most cases the answer is no. But that's where we end.
But you said it's 1 in 50,000, which is 1/3 of the people with phenylketonuria, and those people get diet drinks with special labels saying phenylketonurics beware. So on one hand, for a disease that's only 1/3 as common, there's virtually total ignorance. But
there's two differences here. The one is that while this 1 in 50,000, we only know of about 500 cases globally. The other cases got misdiagnosed as something else. So the community is not that big. Second, we're talking about children that are neurodevelopmentally delayed.
Yep.
So they don't have any power. It's really the families that will have to do the pressure. The kids, they are delayed. They're never part of the society the way we think that they have any power and can apply any pressure. But so CTNNB1, the name sounds a little bit weird, but it's because it's not the name of the disease. The disease is so rare; it's actually not named.
What?
CTNNB1 is the name of the gene that's mutated. So a lot of every single disease I work with, the names are like that. They're symbols. They're just names of the gene, like numbers and letters, and they sound like code, but they are code names for the genes.
So as we're coming to the end, can you just give me a list of some of the other diseases or genes associated with the diseases that you're working with?
Sure. PGAP1, SLC6A1, PUS3, the SLC, there's actually a few different types. They're all codes like this.
And do they affect children in their early years, or do they stay hidden until they become adults?
All the ones I work with, the pediatric neurodevelopmental, they start early on. So that's why the children are the sick one, not later on when they get old. They get affected right from birth, or very quickly the symptoms come up. And we are working, so our patients are actually children, sometimes 1, 2, 3 years old.
So if a parent is worried that their child might have some sort of genetic disease, I guess, what's the first step? To go to their local children's hospital and get a geneticist?
Well, you have to go to, I assume you have to go to your GP and get referred to a genetic, GP? Like a clinical geneticist.
A clinical geneticist?
And do a genetic testing. The technology is getting so powerful that to do a genetic test on a disease, like when SMA was being added to the panel for the blood screen, for the newborn screen, it's about, if I remember correctly, about $7 a person, a test. It's not cheap. If you imagine in this state, 100,000 children get born every year, $7 a test, a new disease. Yeah, just for one disease. So you're not going to test for everything because you can't. Also, do you want to test for something that there's no treatment for? You have to make choices. But now the new technologies such as next-gen sequencing, where you read the whole genome very rapidly and very cheaply, and you have this database. Now you can scan that database for very specific diseases, and the rest of the data sits in the database. If 5 years later somebody comes up with a treatment for another disease, or you can go back now, because the data exists, you just mine the data for another disease, and you can quickly say, over the last 5 years, those are the kids that have this disease, and it costs you nothing. It costs the computer just the power of running the data. So the new technology is gonna really revolutionize this whole field. I don't work on it. I'm going to benefit in the process, and there's a lot of complementary technologies that would work together to make our job more effective, like stem cells, organoid. There's a lot of things that we piggyback off or build on.
So you're associated with the University of Sydney and also the Children's Hospital at Westmead, which is just west of Sydney, and also with Imperial College in London. You were associated with them? Yeah,
so I have my lab is at Children's Medical Research Institute,
Children's
Medical Research Institute,
which is Westmead, which is west of Sydney.
Westmead, the hospital you mentioned, is right next door to us. We work closely with clinicians from Westmead, from Sydney Children's Hospital Network, a CHN. There in Randwick, we are affiliated. CMRI is affiliated with the University of Sydney. I do have an honorary appointment at UCL in London. That's because we have a PhD exchange program where our PhD students can go to UCL and spend part of the PhD there, or the UCL students can come and spend in our lab. So it's a bridge program to allow younger, driven students and scientists to go to another lab, learn something, make connections, build a network, and then go back to the institution. And that's very successful.
So that answers part of my question, do you take students? So if people want to come and work in this field with you, how do they find you and follow you in your fine work?
Yeah, my name is not easy, but it's the easiest way is to Google me or look me up at the University of Sydney website. So it's Lisowski, Lisowski.
And Leszek,
Leszek if you want the written, or Hek if you want to pronounce it.
Or they can look you up at the Children's Hospital.
Children's Medical Research Institute, CMRI.org.au, and I'm one of the lab heads, so that's an easy way to find me as well.
And do you appear on social media as well?
LinkedIn,
Facebook,
Instagram. I don't do Twitter much, but LinkedIn will be probably the easiest one.
Wow! Thank you for opening my eyes. I had no idea that there were so many people affected by genetic diseases. I would have thought it was of the order of 0.5% to 3%, but not 10% of adults and 30% of children. I had no idea.
And what's tragic about that, it's a big market from a perspective of impact, 10%. But there is about 10,000 diseases. So those numbers that like the city and name and the name, there's about 10,000 of those. And some of them, depending on the type of mutation, so depending on what the change in the genome is, they will require different treatment. So you can multiply by probably a factor of 2 or 3 of types of treatments have to be developed. So we need about 30,000 treatments to handle that.
Wow.
So it's very fragmented. It's very difficult. But the way I look at it, people sometimes ask me, why do I work on diseases that have low impact? Because I could work on something common that then I can sell to Pfizer and makes it way easier for me to get funding, to do the work, have global impact. Why do I choose to work on something that may help, 20 kids globally? And the way I think of it, if you're the parent and you have a child with a disease, does it help you in any way if there's thousands of other kids with the same disease or you the only one? Ultimately, at the family level, you have a sick child. It doesn't really, the numbers don't really, strength in numbers, it doesn't always work like that when you are facing a sick child. So I want to make sure that the families that pull the, unlucky loss or the ticket on the lottery of genetic code don't get left behind just because they're not very common or not very, are not united. Every child has the same, every family has the same values and they want to be normal and have a normal life. And I think, that's why the rare diseases.
Look, thank you so much for explaining this stuff that I had no idea of and how you're doing such wonderful work.
Thank you for having me here. I think it's important that people understand that this problem exists. It's, it's not often talked about, but it is a big problem that we're all facing. So.
And with a bit of luck, you'll get some more PhD students.
Yes, please apply. We're always happy to take more students.
Thank you for coming.
Thank you very much.
Shirtloads of Science is washed, spun and aired by the University of Sydney.